Hi. As you might remember, in my last class, I was discussing how viruses infect or specific organs, and before that, it was something discussion about plasma membrane, structure and function. So there are some of the terms when you are talking about fluid mosaic model and selective barrier, selective permeability, all these things, then like first function of plasma membrane is selective permeability. Then there are some important uh, subtopics which you need to know very well, and that is called transport and active and passive transports. So from transport word, you could easily understand what does it mean. So definitely transport here, it does not mean with something vehicle or something. It means transport of some biomolecules from one cell to another or in one cell to another place within one cell. But mostly we are talking about here from one cell to another cell. So passive transport is one of the important biological feature. So as I mentioned, selective permeability in the last class, that means to plasma membrane allows some selective compounds to either pass in or pass out. So this is clear definition of selective permeability. Now, what do you understand with passive and active transport? So passive transport is basically a naturally occurring phenomena where you do not need to supply some kind of energy. So that means it does not require the cell to use the stored energy in any form to accomplish the movement. That means automatically the molecules are moving in or out. That is passive transport. Okay. Then it comes some a term that is concentration gradient, a physical space in which there is different concentration of single substance, which is said to have a concentration gradient. So this will be a kind of important term which you need to know of what is concentration gradient if you want to understand active and passive transport. So here you could see in passive transport, if you want to understand, it may look like something here. So for example, if we are talking about simple diffusion, then it says that the substances or molecules from higher concentrations are passing to lower concentration. That is simple diffusion. Then there is a term called facilitated diffusion, where you could see there is some source or channel via which the molecules or substances are passing. So here then it comes facilitated diffusion in another way. So here it is straightforward. Another is where you allow for from one side to pass, but not to cross something like, carrier protein. Here, for example, it is channel protein. Here it is carrier protein. So facilitated diffusion could take place via multiple source. Simple diffusions does not need any kind of sources, but in facilitated diffusions you need some source like channel proteins or carrier proteins. So these are like some kind of examples. Now in passive transport you could see simple diffusion through the lipid bilayer. As you can see here, this is the same thing. The same thing is shown here. So then it is non-specific transporter. Here facilitated diffusion through a non-specific transporter, then facilitated diffusion through a specific transporter. Then it comes osmosis through the lipid bilayer and an aquaporine. That is what you can see here, aquaporine. Okay, this all. So these are some of the examples, and here you can make some kind of objective questions how the facilitated diffusion could take place. For simple diffusion, you could mostly remember, but for facilitated diffusion, you need to understand some kind of channel proteins, carrier proteins, or some sources, specific or non-specific sources. Now, in facilitated transport, also called facilitated diffusion, materials moves across the plasma membrane with the assistance of transmembrane proteins down a concentration gradient. Without the expense of cellular energy, however, the substances that undergo facilitated transport would otherwise not diffuse easily or quickly across the plasma membrane. The solution to moving polar substance and other substance across the plasma membrane rests or lies in the proteins that span its surface. Material being transported is first attached to a protein or glycoprotein receptors on the exterior surface of plasma membrane, and it allows the material that is needed by the cell to be moved from extracellular fluid. The substances are then passed to a specific integral proteins that facilitate their diffusion or passage because they form channels or pores that allow certain substances to pass through. So the integral proteins involved in facilitated transport are collectively referred to as transport proteins, and they function as either channels for the materials or carriers. Now, there are two types of transport, passive and active transport. In simple definition is in passive transport, you don't need energy. In active transport, you need some kind of energy to take, make it happen. So from higher concentration to lower concentration, this is what you could understand in passive transport, and here in active transport something is going from lower concentration to higher concentration. So this is what you need to remember. Now, active versus passive transport could be seen in another way. So movement against the concentration gradient, as I mentioned, concentration gradient, you need to understand by using energy that is in the form of ATP, and this you could see easily in this way how and where you need the energy to spend. In passive transport, you don't need. So there are two types of transport, simple diffusion and facilitated diffusion. Movement along the concentration gradient without energy is passive transport. That's the easiest definition for it. And this is clear way to understand. I hope that this part is easy to understand. Now it comes at selective permeability. As I mentioned in the last class also, that selective permeability is nothing but to make some molecules to either pass or not that by some based on some selectivity, that is like how cell membrane works. And cell membrane, it is one of the most crucial function of plasma membrane or cell membrane, that is selective permeability. The plasma membrane that surrounds the cell is fluid mosaic of phospholipids like DPPC lipid bilayer and proteins that separates the living cell from its non-living surroundings. The membrane is selectively permeable, consisting of a amphipathic, both hydrophilic and hydrophobic bilayer of phosphate molecules. The layers are held together by hydrophobic interactions of fatty acid tails. So, what is diffusion? Diffusion is a process that refers to the movement of particles from one area of lower concentration. Okay, so sorry, diffusion like higher concentration to lower concentration. So what I was saying basically, when particles are moving from higher concentration to lower concentration area, and then this process leads to equalize the concentration throughout the system. That is very simple way if you want to understand in terms of chemistry. Then it comes some other examples of diffusion through selective permeability properties of plasma membrane. Here you can see how the molecules are moving. And then osmosis. This is something like permeable membrane. You could see here. This is example. So here what it is saying, osmosis is the diffusion of water through semi-permeable membrane according to concentration gradient of water across the membrane. Okay. Whereas diffusion transports materials across membranes and within cells. Osmosis transports only water across the membrane. And the membrane limits the diffusion of solutes in water. So it is a special case, osmosis, of diffusion. Water, like other substances, move from one area of higher concentration to lower concentration area. So this you can see here. In osmosis, water always moves from an area of higher concentration to one of the lower concentration. Now, if you want to compare in terms of figure, diffusion could look like this, osmosis could look like this. Now, another way of presentation, like osmosis, diffusion. So this is the difference. Of course, you can also see somewhere the chart of difference between osmosis and diffusion. Now, tonicity. So tonicity describes the amount of solute in a solution. The measure of tonicity of a solution, or the total amount of solutes dissolved in the specific amount of solution, is called osmolarity. Three terms, hypotonic, isotonic, hypertonic, are used to relate osmolarity of a cell to the osmolarity of extracellular fluid that contains the cells. In hypotonic solution, such as a tap water, the extracellular fluid has lower concentration of solutes than the fluid inside the cell. Water enters the cells. Okay, here, water enters the cell. So in hypertonic solution, so what the fluid contains less water than the cell does, such as seawater. Because the cell has a lower concentration of solutes, the water will leave the cell. So in effect, the solute is drawing the water out of cell. This may cause an animal cell to shrivel or crenate. In isotonic solution, the extracellular fluid has the same osmolarity as the cell. If the concentration of solutes of the cell matches the extracellular fluid, there will be no net movement of water into or out the cell. Blood cells in hypertonic, isotonic, and hypotonic solution take on characteristics appeared in figure 3.22, which was described in the book. But you can see here the example of hypertonic and isotonic hypotonic solutions. Now, another example, this is the figure which I was talking about this here. So how these three type of solutions could look like, and you could understand it in better way. So next part will be that is on active transport. So for today, thank you very much.